Thank you very much. First, I would like to express my big thanks to the committee to give me the opportunity to give this presentation regarding some of my preliminary thoughts on environment and nature study. I'm a little bit nervous here because the last time I had such a nervous is when I was in the US and give my presentation as a tenure track interview. So first I will go to this slide. It give us a brief understanding about population aging, the global and in Asia. First is the annual figure. Here we can see that these skins of these alien peers more and more since about 2018. It could be a little bit more, but you can see that the proportions of the older adults aged more than 60 years old is increasing very rapidly since about 10 years ago. So if you take a look of the Asia, you can see that for China, for example, the proportion of people older more than 65 years old is the proportion is about 12% in 2018, but this number increased to about 15% last year. And the same to the Japan. This is animation here. You can see that the proportions of older increased very rapidly, and due to the low birth rate, people are having a very serious aging issue. And because the population size of the two countries, and as well as other Asian countries, I think Asia is facing a more challenging aging issue right now. So someone say, okay, because the old adults living longer, it could be a very good point because we don't have much more happiness and time with our families, especially the old adults. But unfortunately, based on the study conducted in the US, they found that they analyzed the changes of the life expectancy of the old adults in the US from 1998 to 2006. We see that these proportions increased about 5%, the life expectation. But fortunately, some corresponding diseases like cardiovascular disease, cancers, diabetes, as well as some mortality issues, they increased nearly about 70%. So the thing is, lifespan is totally different from health span. Lifespan is for how long you can live, but the health span is for how long you can live without major conditions. So WHO proposed the health aging, and which means that we can find the process of developing and maintaining the functional ability that enables the well-being of the old adults. So WHO in 2022 also proposed that brain health is very critical for healthy aging. I will skip the definition of brain health because we discussed a lot in this symposium. But if you take a look at the importance of brain health, you can see that the global burden associated with the brain health, like neurological disorders, has been the leading cause of death in the world. But to me, I think because the definition of brain health is still under determination, because we don't have a very exact definition of brain health so far. I read some papers, and people said, okay, we should take a look at the health triangle because it consists of the physical health, the mental health, and the social health. So how about the mental health so far? If you take a look at the burdens of these mental health issues like depression, anxiety, etc., you can see that these proportions of and the burdens of these diseases increase very rapidly, and you can see that especially for middle-aged and older adults, these burdens are quite heavy. And I didn't put a figure here because there is a kind of survey in China, especially in Beijing City. It is the capital of China, but you can take a look at their mental health status of their old adults. It is very unfortunate to see that about 20% of the old adults have the depression or anxiety symptoms, and about 10% of these old adults are suffering from diagnosed depression and anxiety. So I think it's time to synergize the mental health with the brain health. So we have so to me, I think we have a new definition of brain health right now. And if we take a look into the determinants of the brain health, I think we have a lot of top scientists here, and we know that we can do a lot of things to determine the the aging status of our body and even the brain health, I think. So for example, we have the genetic things, we have the social status, and our access to the health care, our health behavior. The most important thing is, and it's also on the topic here today, is the environment. We have the lifetime exposure to the environment, and this also determines our risks to these aging issues, especially the brain health. So how the environment exposures contribute to the aging process. So I give the term here, I call it the environmental aging, to study the aging impact of these environmental hazards. I think it's exposome maybe. So if you take a look at these burdens, so I just give the example that WHO also recognized that air pollution is one of the top killers of the brain health, and based on the most recent GBA report released last 2 weeks ago, so they also said that particulate matter pollution, the PM2.5, is the first killer of the global population. So I'm going to take a look at these associations between the air pollution and the brain health we have here. So the evidence for so far, I think the biological evidence, especially the biological evidences underlying the air pollution and the brain health, is quite limited. So I'm going to answer this research question here. So what is a potential biological mechanism underlying the environmental impacts on brain health? I will take a look into firstly that what kind of or which environmental exposure could influence the brain health of older adults. So we just have a very detailed presentation regarding the air pollution's impact on dementia. So I also did a study about a couple of years ago with Joe here. So we used this normative age study, which is a cohort since 1963, and it was based in Boston. We did this analysis using their cognitive function measurements with multiple visits, and as well as their air pollution status. Now, this air pollution data, they're basically from a fixed monitor in the Harvard campus, and we collected their air pollution data, and collected their cognitive function data. We tried to figure out whether there's any associations between the short-term PM2.5 exposure. So we tried to limit the exposure, the duration, the exposure window of the air pollution to up to 4 weeks, 4 weeks to the 8 days. And we tried to analyze the associations of PM2.5 as well as one of its components, the black atom, with the cognitive functions of these older adults. So based on the mixed linear regression model, we found that all the 2 air pollutants, they are associated with a decline of the cognitive function. But if we take a look into the dose response, and we can see that, there's a non-linear dose response here. And we just take a brief look into the cutoff points. Here we can see that at lower PM2.5 levels, we can have the cutoff point at 10 micrograms per cubic meter. If you found that for these lower PM2.5 levels and these old adults, their cognitive function dropped much more quickly than those exposed to higher levels of PM2.5, which means that the short-term and even lower exposure to PM2.5 could harm your cognitive function. And because at that time we're also interested in about how can we protect our cognitive function, and we thought, the neuroinflammation caused by these air pollutants could be a potential biological mechanism. So we tried to take a look at whether they are taking the non-steroidal anti-inflammatory drugs. Most of them, they took aspirin. So we checked whether they take aspirin could protect their cognitive function. And we can see that for those guys, they took the aspirin, their decline of the cognitive function is much lower than those who did not take it. And if you can take a brief approximately estimation of this reduction, it could be about 70%. So we can have this study here. We have the study said that, we elucidate the short-term impacts of air pollution on cognitive decline of all of these old adults, and we also highlight that lower level of air pollution could harm the brain health. And And we also did some further analysis in the UK Biobank. It's a large cohort study, and we found that even the low-level exposure to air pollutants, not only the PM2.5, but also like the NO2 NOx and PM10, they could also accelerate the development of depression, anxiety of the middle-aged and older adults. So we're gonna have just answer this first question that okay, air pollution could harm our brain health, including cognitive function and depression. So how the environmental exposures affect the brain health? Through what kind of a mechanism? So basically, I'm studying the epidemiology of aging. So so there's a very hot topic in aging epidemiology. We are working on the biological aging. And there's a figure in the Nature paper published in 2019. We can see that if you take a look at this figure from right to left. So we are trying to study those diseases like coronary disease, diabetes, mortality, etc. And they have two causes. The first is aging, and the other thing is the disease-specific risk factors. And regarding aging, we have so many hallmarks of aging. People published like the stem cell exhaustion, the telomerase attrition, methylation, gene damage, etc. And these things, they could be determined by our genes and environment. Because genes, basically, genes is quite fixed and quite stable. So the environment is the only thing that could dominate our aging status, could dominate the biological aging. So I think about whether we could link the environment, biological aging, and those brain health outcomes. So we used this biological age. Biological age, aka this biological clock, is kind of a measurement of biological aging, and I think it's kind of a key to understand the environmental exposures and the human diseases. We have multiple biological ages here, including alerts, like the omics age, like epigenetic age, the metabolomics age, etc. And we also have some clinical biological age based on those clinical measurements, clinical trials. We also have frailty, which we use to determine the functionality of these older adults, etc. So here I used this clinical age in the UK Biobank. We try to figure out whether this biological age could affect the depression and progression of depression and anxiety. So we estimated two biological ages in UK Biobank. First, the Euclidean biological age. The other one is the phenol age. The Euclidean age basically is to try to estimate how long you have lived, and the phenol age is to determine how long you will live. One is for your stability of your system. The other one is for the mortality. And they share some clinical trials here, but basically they are two biological ages for two things. So we also estimated the age acceleration, the differences between these biological ages and chronological ages. We try to understand whether these age acceleration associated with depression, anxiety symptoms or hospital records, like hospital records determined by ICD-9 or 10, and questionnaires, because about 1/3 of the participants, they did kind of a survey about at 7 years, so they're gonna have the PHQ-9 and get 7 measurement of depression and anxiety. And we also have their polygenic research score at the baseline to understand whether these genes, their genetic background, could determine their progressions of this anxiety or depression, and whether the biological age could make some changes on them. And so the first is the associations between these biological ages or age accelerations with the incident depression and anxiety. And we can see that both biological ages were associated with incident depression and anxiety separately. But if you take a look, because depression and anxiety, they're usually coincident together. So we usually do, we try to figure out whether the phenol age or the Euclidean age acceleration was first associated with the coincidence of the two diseases using this model. And we found that, okay, the phenol age is still associated with the comorbidity of depression and anxiety, but unfortunately Euclidean is not. Which means that, okay, because the Euclidean was for the stability of your organism, which means they're not tightly associated with the mortality, and phenol age is very closely associated with mortality, and the coincidence of depression and anxiety is highly associated with mortality. So we make these assumptions here. And we take a look at these syndromes of depression and anxiety based on the questionnaires. Their baseline aging status, their associations with these syndromes happened 7 or 8 years after this baseline survey. We can see that we can see some of these syndromes, they are associated with the two biological ages. And the last one is the GBAE. I'm not saying that GBAE, but the gene background and biological age. You can see that if though for those biological aging and social risk of depression and anxiety, this could be enhanced by the genetic risks of these participants. So based on these studies, I think cooperating with the studies I described in the first part, I think we did some analysis, actually 3 analyses in the UK Biobank. We found that we linked the environmental exposure with biological aging. We used the air pollution. On air pollution could accelerate your biological aging. And secondly, that we found that air pollution could harm our brain health. And we also in this study found that biological aging could link the harms of long exposure with brain health. So the 3 studies together suggested that okay, biological aging could be a potential risk factor for brain health and potential target for the risk assessment and intervention. And we also did kind of another studies because the multimobility is also another issue of the aging population. We did these studies and compared with some traditional scores. So this study basically is for the accelerated biological aging with the cardiometabolic multimobility, which is defined as you must have at least 2 of the 3 diseases, the IHD, the stroke, and type 2 diabetes. We found out whether the progressions of this multimobility associated with your baseline aging status. And we can see that, okay, the aging status at the baseline could predict their transition probability and their risk at each stages of the cardiometabolic multimobility. So in together, I think in terms of depression, anxiety, or stroke, we found that okay, the biological age is kind of a link to the brain health. And the third question would be, what is the potential biological mechanism? So I did a lot of studies of DNA methylation. So I think I'm gonna move to this figure because the time is limited. And the first is that for the DNA methylation, people have done a lot of good systematic, good reviews regarding the hallmarks, especially the biological hallmarks. We found that hallmarks of aging, hallmarks of environmental source, just to put them together, we can find some overlap. Some overlapped hallmarks here, like the first one is the epigenetic alterations. And one of the epigenetic alterations is the DNA methylation. It's kind of a, how can I say, I borrowed this from my supervisor, Andrea Petterelli. It's like a pencil marks on a printed notes that it could change the sound you heard, but it did not change the notes itself. So basically, based on previous studies, DNA methylation opens the gate for us to study the underlying mechanism of environmental aging. So I think I could go to the third part of the presentation. This study is about air pollution and epigenetic aging. It is kind of a quasi-experimental study. We collected about, actually these 26 college students, and then asked them to collect their blood samples before, during, and after PM2.5 wave in Beijing, because Beijing at that time in 2018 and 2019, air pollution were still quite serious there. And we tried to figure out whether their demethylation profiles, including the epigenetic ages, could change in these short waves of PM2.5 pollution. We collected blood samples, as I said, and measured the demethylation profiles of their CD4 cells using the Illumina EPIC chip, and estimated 7 epigenetic ages. These just some examples of these biological ages, like demethylation, mortality score, and pace of aging, etc. And we try to estimate it, the associations of these PM2.5 and these 7 epigenetic ages. The first figure on the top left is PM2.5 distributions during this period. We did these studies in 4 batches, And in each batch, you can see that the PM2.5 levels is quite high. And it is kind of a PM2.5 dominant scenario, exposure scenario is dominated by PM2.5. And we use with this design, we obtain kind of this scenario and try to figure out whether whether these changes of this PM2.5, I mean the short-term exposure, only about 48 hours, and could accelerate these biological ages. And we found that 6 out of the 7 epigenetic ages, there are changes. I mean, the differences between the 3 time points could be accelerated by the short-term PM2.5 exposure in 24 hours. And we just expanded this exposure window to 48 hours. There are still 3 epigenetics could be accelerated. So this is for this study, and we just further take a look into all these inflammation biomarkers and oxidative stress biomarkers using the mediation analysis. You can see that some of these circulating biomarkers, they could mediate the impact of PM2.5 on the 3 biological ages. So we can see that, we suppose that, okay, we made a conclusion that the short-term PM2.5 exposure could accelerate biological aging, reflected by the DMS gene profiles. But the thing is, this is not the end of the story. So we further, because DMSation, it is omics.
Sorry to interrupt. You know, those who want to catch the first bus, just go there, okay? The first bus is waiting. So, and then we can continue until 5:30 if you want to, stay here.
So this is not the end of the story. So we try to, because all this data, you need kind of a validation using unique samples, using independent samples. So we find another quasi-experimental study in China, and we try to figure out whether whether the DMSing changes of some of the genes could mediate the impact of PM2.5 on these biological ages. So the first thing come to my mind is the sleep-related genes, the circadian-related genes, because in our previous studies we found that air pollution could harm your sleep quality and then further accelerate biological aging. So to validate this theory, this hypothesis, I think we used these data from the 2 experimental studies. We tried to figure out whether about 1,200 city sites located on the circadian genes, their changes could mediate the impact of PM2.5 on these epigenetic ages. And we found 3 city sites on these genes could mediate the impact. And some of them, they are associated with PM2.5. 3 of them associated with PM2.5. They are also mediate the impact of PM2.5 on the epigenetic ages. We did kind of a high-dimensional mediation analysis. We found 2 of them, they mediate a good amount of proportions on that, which is around about 10%. So in short, I think we have 2 clocks in our body. The first, the biological clock, determines our lifetime or health span, and the other one is the circadian clock, determines our 24 hours activity. And these 2 clocks, they have some links with each other. They could be affected by the external exposure, like air pollution. The last slides of this presentation could be the scheme of environmental aging. So we did a lot of study on environmental exposure as well as exposome, like air pollution, metals, smoking, etc. They are associated with aging health outcomes, including brain health. And we try to figure out whether this biological aging play a very important mediating role here. We can use these omics data like epigenetic, genetic, or even, as we discussed here, with these exposomic data. We want to figure out whether if these, using these data or use these findings, we could make some precision intervention or precision prevention on these impacts of environmental exposure on these aging outcomes. We try to implement these omics data to uncover the individual's biological aging status, to validate their vulnerability and corresponding risks of aging-related health outcomes. That's the end of my presentation, and special thanks to my supervisor, Andrea, and Joe here. Thank you very much. Andrea is now you cover, but still, I did most of my work when I was in North America. is from Peking University, China CDC, and final support from NSFC, the National PID Program of China, and NIH.